good afternoon to everybody. I say that with the caveat that the sun has just risen here in northern Colorado. So the project I'm going to talk to you about today is perhaps sort of a quintessential COVID project. For the last mostly the last 2 years, we've had the ability to get out in the field and collect samples, and we've had continued access to our servers and our bioinformatic pipelines. But less access, due to lockdowns and everything else, to our wet lab facilities. And so what I'm going to talk to you today about is more of a genome-centric view of microbiomes and how they interact following wildfire, which is obviously a huge concern here in the Western US. Before I jump into the results, I just wanted to acknowledge the people who have actually done the work. So Amelia Nelson is a graduate student in my group in the Soil and Crop Sciences department, and she's done the vast majority of the analyses that I'll show today. We also work closely with Chuck Rhoades, who's a biogeochemist at the US Forest Service here in Fort Collins. Holly Roth is a PhD student in the chemistry department at CSU, and Thomas Borch is a soil chemist at CSU. The work is supported through the National Science Foundation and USDA NIFA, and we also have support from the Joint Genome Institute and EMSL as well. So I want to start out first by just highlighting the importance of forested ecosystems and forested sort of high elevation catchments, especially in the Western US. But at a global scale, the ecosystem services that are provided by forests are astronomical. They're estimated at about $5 trillion annually. And like I say, I'm here in Colorado, so this is going to have more of a slant towards the Western US. But the high elevated high elevation forested watersheds that exist in Colorado are critical to the existence of populations in the Western US. They provide drinking water to over 30 million people and irrigate about 4 million acres of arable land. Of course, forests play critical roles in biogeochemical cycles as well. And if we just want to focus on the carbon cycle, globally again, forests store about 860 gigatons of carbon in both live and dead biomass, and also in soil organic carbon. And temperate forests in the US are actually the largest land sink of carbon in the country and offset a pretty significant fraction of the US's CO2 emissions. Now, talking to hopefully a sort of a microbially focused audience, and so it should come as no surprise that microbes in forest soils catalyze myriad critical reactions. And so this was just a nice figure from Noah Fira a couple of years ago, just really highlighting the critical roles that microbes in soils play in cycling everything from carbon and nitrogen to sulfur, iron, manganese, and a wide variety of additional elements as well. So microbes play critical roles in biogeochemical cycles in soils generally, but also forest soils. And perhaps just as critically in forest soils, microbial symbioses with plants in the rhizosphere are often critical for the establishment and persistence of trees. And so we just have an image here of lodgepole pine seedlings, which are very prevalent in the southern Rockies. And here, these trees rely on symbioses with ectomycorrhizal fungi in the rhizosphere to actually to grow effectively within the forest. Now, as critical as these forested ecosystems are, they are also at risk of massive disturbance. So this is a photo that was taken just south of my house in Fort Collins in September 2020. And this is the Cameron Peak wildfire, which was just starting to blow up then. And it ended up being the largest wildfire in recorded Colorado state history. And that summer, this wasn't the only wildfire in some of these forests of the Southern Rockies. This is just an image from of northern Colorado, and you can see the Cameron Peak fire there, but there were also huge fires, the East Troublesome fire, the Mullen fire, and some smaller fires dotted around as well. So we saw a huge fire season linked to changing climate, forests are drying out, and we're also dealing with sort of historic management practices in these forests as well. And this sadly wasn't just a Colorado problem as well. This is data across the US showing annually the number of areas burned by wildfire. And you'll see the general trend is for an increase over time. Additionally, this is not just a US problem as well. I'm sure many of you remember just this past summer, catastrophic wildfires in Greece and Turkey. And more recently in the news, we've seen wildfires in Australia, out in Siberia, and in the Amazon as well. So we're seeing this increase in wildfire across the globe. And importantly, we're also seeing changes in the severity of wildfire as well. So many forests are adapted to fire. Fire is a component of healthy forests, especially here in Colorado. But traditionally, over the evolution of forests, they're used to seeing sort of low to moderate severity wildfire. But again, as forest management practices have changed and our climate has warmed and forests have dried out, we're now seeing increasing amounts of high severity wildfire. And so the image you're looking at here is the burn severity map for the Cameron Peak Fire, which was the fire just west of us here in Colorado. And the red areas indicate areas that burned at high severity. And so traditionally, these forests haven't been burned to this extent before. And so our understanding about how the forest will respond and how the soil will respond to these high severity fires is really in its infancy. So how does wildfire impact soils? And this in of itself could be a long talk, but I'll keep it short just to a couple of bullet points. Obviously, during wildfire, we see a lot of combustion of vegetation, and so we see loss of vegetation. We also see increases in soil pH. The organic matter that binds soils together and creates aggregates is frequently lost during combustion, and so we lose a lot of soil secondary structure. And for the organic matter that remains in the soil, we also see changes in the structure of that organic matter, and I'll get to that shortly. But perhaps most importantly for this presentation today, we also see combustion of microbial biomass as well. And there was a really nice sort of meta-analysis synthesis that Yamina Pressler did a couple of years ago. And across hundreds of studies, she deduced that fire reduced biomass and abundance of microorganisms by up to 96% in surface soils. So these catastrophic disturbances have the significant role in removing microbial biomass. So with this in mind, we went into this study asking sort of three key questions. The first one is, how does wildfire alter the composition of the soil microbiome? So in the aftermath, when we've lost all this biomass and all these available niches are open within these soils, which microbes are able to come in there and colonize those soils? The next logical question is, which strategies do microbes actually use to do this? How do they tolerate these sort of somewhat extreme conditions in the aftermath of wildfire? And then finally, what are the implications of these changes? Do we gain or do we lose any key functions associated with the soil microbiome in the aftermath of wildfire? So these are the three questions that we wanted to investigate. And so to do this, we focused on two fires. And these were two fires, the Ryan Fire and the Badger Creek Fire, that actually burned in 2018. They burned right on the Colorado-Wyoming border. And what we did was we went out to these locations about 9 months after these fires had been extinguished. And we identified 4 burn severity gradients. And so that's the image that's just shown on the right here. You can see we're going from an area that's burned at very low severity. So there's still some green in the trees. There's still a lot of organic material on the surface. And then we're getting into areas of high severity. And that's the really blackened area you can see at the end of the arrow, where we've basically combusted all the organic matter on the soil. We have 100% tree mortality. So we went to each of these 4 burn severity gradients. And so these are just some closer up images so you can get an idea of what the soil, what the forest floor actually looks like. And at each of these, we collected the surface layer. So obviously in the control and the unburned and the low samples, there was a legitimate O horizon that we could actually sample. Once we get into the moderate and the high severity soils, that O horizon has been combusted. And so we're really sampling the ash layer at that point. And then we also sampled the deeper mineral soils, the A horizon soils. Once we had collected all these samples across these 4 gradients, across these 2 fires, we then applied a suite of different analyses. We used mass spectrometry to try and identify the pool of dissolved organic matter within these soils. And then we also applied a suite of microbial analyses. We did 16S and ITS sequencing to really characterize who's there. We applied metagenomics to see which genomes we could reconstruct and what the functional potential of the community was. And then we also used metatranscriptomics to look at the RNA, the messenger RNA that was being produced by these communities, to start to get an idea as to which genes are actually active and what the microbes might actually be doing in these soils. So first of all, we'll start off with just sort of the who's there and how did the community change at the compositional level. And as expected, I've already talked about how we lose a lot of biomass and we transition to these somewhat extreme conditions characterized by high pH and whatnot. And so very clearly trend in the organic horizon, we see significant decreases in the diversity of both the bacterial and the fungal communities. So here we're looking at just one measure of alpha diversity, Shannon's H. And you can see that as we go from control to low to moderate to high severity impacted soils, we see that concurrent decrease in the diversity. But interestingly, once we get into the mineral horizon, just a few centimeters below the surface, those effects are far more muted. And so while we do still see some decreases in the bacterial diversity, the fungal community diversity doesn't actually change very much at all. So the takeaway here is that these deeper mineral soils are actually somewhat insulated, at least in terms of diversity changes from the effects of wildfire, even under high severity impacts. In terms of who actually changed in the community compositions, probably the clearest signal, and this perhaps isn't unexpected, was this huge enrichment of actinobacteria. So what this graph is showing is just percentage change under different burn conditions relative to our control and burn soils. And so in that first series of columns, you can see that with increasing burn severity, we actually saw increasing enrichment of taxa affiliated with actinobacteria. And we see similar trends for, but less large for things like chloroflexi, proteobacteria, and Bacteroidetes. But the other sort of clear trend here is we see a loss of many soil microbial taxa, and those are the groups on the far right side of this image. We can see that many of these groups decrease almost 100% with moderate and high severity wildfire. Okay, so that tells us how the alpha diversities change. We've got some insights into how the composition of the communities changed under wildfire. But of course, we're interested in what the functional implications are of these changes. And so here's where we started to apply some of these sort of multi-omics tools, metagenomics and metatranscriptomics. And we performed this sequencing on a subset of samples. So we did this sequencing on low and high soils from the organic and the mineral soil horizons. And specifically from the metagenomics, we were able to recreate a fairly large number of genomes. So through our computational pipelines, we were able to reassemble approximately 640 bacterial metagenome assembled genomes, or MAGs, just a couple of fungal genomes, about 2,500 viral genomes. So we have a pretty good representation of who's there, and that's shown in this graph on the right. Based on that compositional 16S data I just showed you, it was reassuring to see that the highest number of MAGs that we were able to reconstruct were affiliated with the actinobacteria, but then also proteobacteria, Bacteroidetes. And you'll remember those are the taxa that we did see responding positively to fire in the previous slide. So we have this database of genomes now, so we can start to probe and see what functions do they encode, how might they be able to colonize these burned soils. And what we saw there were a series of what we infer to be critical traits encoded within these fire-responding microbial genomes. So the first one, perhaps not unsurprisingly, was some level of thermal tolerance. And so this graph here just shows the change in gene coverage as we go from low severity fire to high severity fire. And you can see, for example, sporulation in both the mineral and the organic horizons. We see an increase in the abundance of genes associated with sporulation pathways. And the same goes for heat shock proteins as well. So it seems like the community, as we transition from low to high severity fire, does encode more genes associated with thermal tolerance. And so, like I say, perhaps not unexpected, but the first critical trait is this ability to persist through disturbance via thermal resistance mechanisms. I also want to talk briefly for another trait about growth rates. And so this was a nice paper that was published earlier this year in ISME out of Jed Furman's lab. And they took this concept where we can look at the frequency of nucleotides within genomes and actually use this to predict maximum doubling times or growth rates of microbes from isolate genomes or from metagenomes. And so the graph on the right here just shows a study they did where they predicted growth rates based on genomic data, and then they actually performed experimental studies and grew the same microbes in the lab and measured their actual observed growth rates. As you can see, there's a nice positive correlation there. Obviously, it's not perfect, but it's enough to be able to start to infer growth rates from simply genomic data. So that's what we did with our MAG database, our 640 bacterial genomes. So what we're looking at here, I'll just spend a second to actually explain what we're visualizing. On the y-axis, we're looking at the doubling time. So as the dashed line is the database average. As we get below that, these are genomes with the potential for the organism to double faster. And above the dashed line are organisms that would potentially grow slower. And as you can see, on the x-axis, we also have the MAG relative abundance from 0% all the way to 6% relative abundance. And the trend is most clear in the soils impacted by high severity fire, which is the top graph here. And the trend here is that those organisms which are most abundant in the soils 9 months after wildfire also have the potential to grow faster. That trend is also somewhat apparent in the O horizon soils impacted by low severity wildfire, which is not quite as significant. But then interestingly, when we look at the A horizon, again, where we would expect to see less biomass loss and less impact of wildfire, those trends are not apparent at all. You can see there's no real significant trend here at all. So our takeaway here is that where we've had high severity fire, we've lost a lot of biomass, we've opened up a lot of niche space in the soil. Organisms that can rapidly grow, have the potential to grow rapidly and fill that niche space in the aftermath of wildfire, have a definite benefit. Now, let's talk briefly about chemistry and the formation of pyrogenic organic matter. So we did some very simple water extractions to extract the pool of organic carbon, which we thought microbes may be able to access. And then we analyzed this using mass spectrometry at EMSL in Washington State. And this is just a somewhat messy overview of the image here. I'm sure many of you are familiar with Van Crevelen diagrams. This technique this way of plotting the data is just by plotting the hydrogen to carbon ratio versus oxygen to carbon. And what we're looking at here are the unique compounds that we detected in control soils impacted by low severity wildfire, and then we grouped moderate and high severity wildfire together. And there are some trends here. Hopefully you can see that those red circles are definitely trending down towards the aromatic line that we've drawn in this graph here. But a far simpler way of displaying this is simply by considering the aromaticity index of the organic carbon that we've measured using mass spectrometry. And this is a very clear trend. You can see that even the low severity conditions going all the way to high severity wildfire conditions, we see this increase in aromaticity of the soil organic matter. So the question at this point is, well, can microbes access this dissolved organic matter that has this increased aromaticity? And of course, there are thousands, tens of thousands of different aromatic structures that are generated during wildfire, and the gene pathways are not well annotated for understanding how they're processed. And so instead, we focused on two intermediates of those degradation pathways. And so that's what this diagram is showing here. Catechol and protocatechate are both intermediates in the degradation of diverse aromatic compounds. And we're actually able to study the degradation pathways for catechol and protocatechate. We know what the genes are, and we're able to identify them in our genomic data sets. So again, that's what we're looking at here. We're looking at the degradation of catechol and protocatechate all the way to TCA cycle intermediates, succinyl-CoA and acetyl-CoA. And we were able to detect all of these genes across the genomes that we recovered from these burned soils. And then when we look at actually the expression of these genes, we again see that these arrows just indicate we're detecting transcripts for each of these genes. And you can see that under both high severity O soils and high severity A soils, we do see expression of all of these genes leading to the production of succinyl-CoA and acetyl-CoA. I'll just show one sort of final figure here that's probably not super clear, but when we look at who is actually expressing these genes, it's the microbes that we might expect. In the O horizon soils, we see a lot of actinobacteria that are expressing partial parts of this pathway. And in the A horizon, again, we see a lot of actinobacteria there, but we also see some increased diversity there, some proteobacteria and other tacta that are contributing to the degradation of these aromatic compounds. So our takeaway from this analysis here is that Another key trait that allows certain microbial taxa to persist in these samples is the ability to generate wildfire-generated pyrogenic carbon substrates. So I'd also like to talk briefly about the potential role of viruses in these systems as well. Now, as I'm sure many of you in the audience are aware, viruses can infect microbial cells. They latch onto the exterior of the cell. They inject either DNA or RNA into the cell. They hijack the host, the bacteria or archaeous machinery, and make many copies of themselves until the cell effectively bursts. And this is what we're looking at here. This is just an image that we took a couple of years ago of one of these microbial cells bursting, and you can see both viruses in this image leaking out of the cell, but you can also see cellular contents. So all that tasty organic carbon that resides within a microbial cell is also leaking out as a result of this cell lysis. And so in all these ecosystems where viral predation and viral induced cell lysis occurs, we infer that the release of this organic carbon represents a labile carbon source for the other microbes in the system. So we looked for viruses across our genomic data sets, and we were able to detect both DNA and RNA viruses. We saw about 2,500 distinct viruses, and approximately 1,000 of these were novel, so they hadn't been seen in any other data set, whether it be soils or the deep subsurface. A critical part of this is then actually saying, well, we have these 2,500 viruses. Which microbes might they be targeting? Which microbes might they be able to infect? And we can use a sort of a suite of computational tools to start to get at this question. So what I'm going to show you now is a graph. And again, on the x-axis, we're looking at the relative abundance of these different genomes. And on the y-axis, we're looking at the number of different viruses that could potentially interact with that genome. Okay, so hopefully the takeaway here, and again, we're looking at samples from O-horizon soils impacted by high severity wildfire. And so the trend here is basically the genomes that are most abundant, so between maybe 3 and 6%, also have a relatively high number of potential viral interactions or viral linkages. This trend is also apparent, again, in the deeper A-horizon soils. And so what we're seeing here is sort of what we call a kill the winner strategy. The microbes which are doing better in the soils are targeted by more viruses, which again leads to increased lysis of cells and potential release of organic carbon. And this isn't a trend that we just see in microbial ecology. This is a trend that's commonly seen in macroecology as well. It's simply predator-prey relationships as highlighted just in this simplified graph here as well. So to wrap things up here, I just want to talk briefly about the absence of vital ecosystem functions. We've talked about which microbes can colonize these soils and how they can colonize. So I want to just talk about what's missing in this sort of new microbiome in the aftermath of wildfire. So I mentioned at the start that we see a decrease in fungal diversity with increasing burn severity. And critically, when we look at the types of fungi which we actually lose, we see that we lose almost all of our ectomycorrhizal fungal species. And if you remember, these are the species that are key symbionts of lodgepole pine. And this is kind of concerning because if you imagine in the aftermath of wildfire, you're expecting new seeds to be able to colonize the soils and start to germinate. And they require the presence of ectomycorrhizal fungi. And it looks, certainly under high severity conditions, like we're losing massive amounts of EMF. So the question is, how does that impact the germination of lodgepole pine seedlings? And that's a question for another day. We haven't talked specifically about the nitrogen cycle just yet, but the nitrogen cycle is really important in forest soils, especially after wildfire. We see a lot of soil nitrogen loss during wildfire. And so you would expect that nitrogen fixation would be a really important process occurring 9 months after wildfire. Despite this, we didn't really see any evidence at all for the expression of NIF nitrogenase genes, which actually perform nitrogen fixation in these soils. And so it's currently unclear whether this is simply a sampling artifact or why we're not seeing evidence for this critical process in these forest soils. Finally, the results also have implications for the stability of pyrogenic C. This carbon is typically thought to be a recalcitrant to microbial degradation. And so it's thought to be sort of a relatively stable carbon stock in the aftermath of wildfire. And yet we see the functional potential and the activity for pyrogenic DOM degradation across a large number of genomes that we recovered, both bacteria and fungi. And so, again, this is ongoing work, but this, of course, has implications for post-fire carbon storage in these burned soils. So to summarize, increasing wildfire severity drives decreases in the diversity of both bacteria and fungi in these soils. Actinobacteria are by far the most enriched group of microbes in these soils, and we think they're able to dominate this ecosystem because they produce things like spores, they can grow fast, and they have the potential to utilize pyrogenic carbon substrates, which are abundant in these burned soils. We also see abundant viruses, and so we've sort of inferred this potential role for viral predation and cell lysis as another mechanism that's releasing carbon into these soils and is stimulating microbial activity. In terms of other processes, we see this loss of ectomycorrhizal fungi, and so we're still investigating what the implications are for the reestablishment of trees in these burned landscapes. And then we also see this massively disrupted nitrogen cycle. And so given the loss of nitrogen from burned landscapes, we're particularly interested in how this nitrogen cycle reestablishes itself and develops sort of moving beyond that 9-month period. So I think I'm back to 8 a.m. my time, which I think gets us on track still. So that's all I have for today. If anyone's interested in this, we have a preprint out at bioRxiv right now, which people can access. But yeah, thank you all for listening. I appreciate it.
Thanks, Mike. That was a really interesting talk. Thank you very much. Does anyone have any, let me just look at the chat. So we have a couple of questions here. So hi, Mike, fantastic talk. Playing with wildfires, quite cool research areas. I'm just wondering, how can you track the microbes' origin from after the wildfire? They recover from deeper soils or mainly from aerosols? Does the aerial virus particulates migration matters to the shape of the microbial community?
Yeah, that's a fantastic question, and actually something that we spend a lot of time pondering. So I think we don't really know right now. I suspect many of these microbes do come in sort of on aerosols from the atmosphere. Some, I'm sure, migrate from deeper depths that are more sort of thermally insulated. And of course, those that are present as spores probably stand a greater chance of persisting even in some of these burned areas. But to start to address this, obviously, it's kind of difficult to address this in the field, because usually when there's a wildfire burning, we're not allowed anywhere near it. So we have experimental apparatus now, which allows us to actually sort of combust soils in the lab, and then we can take them out into the field, and we can start to get at this sort of immediate colonization question. We can sort of bury these soils in the forest just west of us and start to infer, are things migrating from deeper, or are they coming from the atmosphere? So that's a great question, and hopefully, in a year or so's time, we'll have a better idea of that.